Bienvenidos todos. Vamos a iniciar esta conferencia internacional. Antes que nada, quiero manifestar un agradecimiento al apoyo oportuno, amplio que nos ha dado la doctora Medina-Mora para la realización de este evento, en primer lugar. En segundo lugar, a la Asociación Mexicana para el Estudio y Tratamiento del Dolor, y en particular a su presidente, el doctor Ángel Juárez, que nos compartió y nos apoyó para poder invitar a la doctora Luana Colloca de la Universidad de Maryland para que estuviera con nosotros y nos compartiera parte de su riquísima experiencia de investigación en el campo de los mecanismos del placebo y del nocebo. Bueno, nos acompaña en el día de hoy la doctora Medina-Mora, directora de nuestra facultad, la doctora Magda Campillo, jefa del área de investigación y posgrado. También nos acompaña la doctora Laura Hernández, del área de Psicología Clínica y de la Salud, el doctor, nuestro querido colega de la UAM Lerma, el doctor Gerardo Pacheco-López, un experto también en el campo del placebo, y acompañando para fines de traducción en cortito, Jairo Domínguez, que nos va a ayudar para que la doctora no se pierda nada de lo que se habla en español en esta sesión. Vamos a darle la palabra a la doctora Medina-Mora para iniciar después con la conferencia.
Muchísimas gracias por organizar este evento, por tener el honor de tener aquí en esta mesa a figuras tan importantes, a tratar el tema que vamos a tratar hoy, que es un tema que a todos nos compete, porque todos en algún momento necesitaremos manejo del dolor y además, pues en nuestros países, en vías de desarrollo, tenemos poco acceso a los medicamentos. Más o menos es el 12% de la producción de las Naciones Unidas, por ejemplo, de morfina, que se queda en nuestros países, y los demás en los pocos países de nivel alto de desarrollo, cuando en nuestros países vive el 80% de la población que lo necesita. Y estas alternativas que desde hace muchísimo tiempo nos ha planteado y nos ha enseñado el doctor Benjamín Domínguez, pues ciertamente son muy importantes. Me ha tocado el honor de ver a sus pacientes, de lo que platican, de lo que han aprendido. Y bueno, todos sabemos que la parte emocional del dolor es una parte que podemos atender y que podemos controlar de tal manera, bueno, no controlar totalmente, pero avanzar en pues, hacerla positiva. Y pues también pues, el gran honor de que podamos tener a la doctora Luana Colloca, perdón, me equivoqué, Colloca, con nosotros, que es una distinguida profesora de la Universidad de Maryland y profesora honoraria de la Facultad de Psicología de la Universidad de Sydney, y pues ya nos darán su semblanza, y estamos muy, muy agradecidos que estén aquí con nosotros. Tenemos con el doctor Sánchez Sosa en Psicología de la Salud, un programa de cuidados paliativos en esta explicación amplia del momento en que la vida se pone en compromiso, que puede ser muchos años antes de morir. Y pues los psicólogos hemos trabajado además en cómo ayudar y enseñar a las personas a morir, cómo ayudar a la familia, cómo dar las malas noticias, como decía el doctor Sánchez Sosa. Creo que ese trabajo conjunto que tenemos ahora de médicos y psicólogos de nuestra facultad hermana de medicina aquí en la universidad, pues ha sido muy provechosa para el trabajo en los hospitales. Así que muchísimas gracias, Benjamín, por esta gran oportunidad y gracias a todos ustedes que estén aquí.
Antes de darle la palabra a la doctora Luana, quiero mencionarles que con el apoyo del área tecnológica de la UNAM, estamos literalmente estrenando en esta facultad la aplicación de Zoom para generar los subtítulos en español en tiempo real de lo que nos va a compartir la doctora Luana. Y eso realmente fue una distinción que nos hacen a la Facultad de Psicología de estrenar esta aplicación. Entonces no se preocupen por la traducción de lo que ella nos va a presentar en inglés. Se van a generar aquí los subtítulos en español en automático y espero que todos lo usemos después de esta ocasión. Entonces no se nos va a perder nada, ¿sí? Adelante, doctora. You prefer to talk here?
I wish to express my gratitude to Dr. Medina-Mora. Thank you very much for organizing, together with Benjamín, next generation of scientists and the next generation of physicians treating pain. And also it's my great pleasure to meet again Gustavo. He was a pioneering researcher when I was a PhD student together with Shadilowski, publishing among beautiful, elegant papers on modulation of placebo effects through neuroimmunology. So I hope you will read these papers and take the opportunity to talk to him. And so today we talk about pain, management of pain without any pharmacological treatment. May sound challenging or also a paradox. I'm a physician scientist, not a psychologist, but I believe that we need to integrate psychology with pharmacological approach to pain. That will be the solution to treat both the intensity and the emotional component of pain, what we call affective component of pain. And so today the goal is to learn more about the neurobiologic mechanism of placebo analgesia with an overview of some pharmacological study, social learning, that is something that we spend time now in the lab. And the second part of the talk is focused on results obtained directly in patients suffering from chronic pain. Sometimes I like to challenge my audience and ask, what do you think placebo effect is? So maybe I can ask, right? Anyone would like to volunteer to answer, what is a placebo, what is a placebo effect? And you can reply in Spanish language if you prefer. Anyone would like to comment what a placebo is? Oh, no, you. Great. So a placebo is an inert treatment introduced in medicine to compare treatment we want to validate, like a pharmacological treatment, with an inert treatment. And usually the shape, the smell, the color is identical to the active treatment. And sometimes this becomes more challenging. For example, if we want a sham surgery or a sham acupuncture. Acupuncturists were clever in creating this needle that retracts, or sham surgeon cut the skin, do still the debriefing, the cleaning of the wound, but they don't do the procedure that is meant to create the treatment, for example, for vertebroplasty. So the goal is to understand that no matter which treatment we use, a pharmacological treatment, a surgical treatment, I have here a vertebroplasty, or, for example, integrative medicine, always, these treatments are not provided in a vacuum, rather in a psychosocial context, the bedside, the clinician, the patient realm, life, prior experience. So if we use opioids, here is morphine, vertebroplasty or needles for acupuncture, always we have a combination of the specific pharmacodynamic component plus the expectation component. So our mindset, our expectation. So if we try to separate these two phenomena, I will show to you that things get tricky. And to demonstrate that placebo is part of any treatment, we create this very inexpensive paradigm. I was still a PhD student in Italy, and the resources were limited. So we try to compare the response to medication provided to an IV line versus a situation where a physician is at the bedside and tell patient, now you are going to be treated. So we want to compare merely adding the clinician, the nurse, or the doctor there, how this can change the outcome. So, and I'm lucky because the translation doesn't cover my slides completely. So we have 4 different treatments, buprenorphine and tramadol, that are 2 opioids class of medication, ketorolac and metamizole, non-opioids, what we call NSAIDs. So the goal was to compare the same identical dose of pain therapeutics when we provide the drug in a hidden fashion versus an open fashion. Here patients receive through an IV. They knew that they are treated, but there wasn't this empathic presence, or even not even empathic, a person to tell, I'm here for you. And this is the interaction, what we call therapeutic alliance. And so, what I would like to stress here, anytime we provide a drug in a psychosocial context, there is this placebo component. So here I show to you that we can study placebo effects without any placebo. And no matter which drug class we use, opioids, non-opioids, always the placebo component boosts the action of the drug we are using. After 20 years from this research, we know much more about the placebo phenomenon. For example, the goal was, how can we induce these effects in patients, in humans? So, well, we can use verbal suggestion. This is a powerful treatment. We hope you will be feeling better. Or we can use prior therapeutic experience, a conditioning, where we treat a patient, for example, for many times with NSAIDs or morphine, gabapentin, and then we replace the treatment with a subtherapeutic dose that will count as placebo. Or we can also learn from other people and the therapeutic alliance with the patient. All these are somehow psychological pathways to induce an expectation of placebo analgesia, pain reduction. And after 2 decades of brain imaging studies, we know that through placebo effects, we can modulate the transmission of the pain signaling, what we call nociception, to the brain. And this modulation can go in both directions. Positive, that means we somehow amplify, and that is the nocebo component, or negative, we inhibit the transmission of pain. And there are parts of the brain that are super critical in induce an expectation of analgesia, like the dorsolateral prefrontal cortex, the ventromedial prefrontal cortex, the nucleus accumbens, the ventral medial. So these parts of the brain are those that make us humans. The reward system, seeking pain relief, but also then we have part of the brain in red, like the anterior insula, the dorsal anterior cingulate cortex, the somatosensorial area, that allow us to say, I have pain in my knee, the pain is 8, 5 of 10, and it's a sharp pain. That is the reason why patients describe so beautifully their pain to us. And finally, nature gives us some part of the brain to modulate this transmission of the nociceptive signaling, the periaqueductal gray, the spinal cord, and the rostroventral medulla. through that area, we can amplify, like when we press the accelerator to come here and be on time for the lecture, versus braking to avoid an accident, right? The same, our brain has this powerful area to either press and feel more pain, that is a catastrophe for our patient, or can be also protective in some circumstances if we need to retract the hands from fire or from burning ourselves while cooking, versus, inhibiting to feel relaxed, to feel like, okay, this pain is in my hands, I can handle it. The nocebo effects is somehow the negative counterpart that make us feel more pain when in reality the level of intensity of a painful stimulation is not so high. And this occurs in many patients for different factors. We have some internal factors like catastrophizing, mood, emotion, maladaptive cognitive appraisal, patients who had the trauma during or who grow up in situations with disparities, low income, low education, and so on, versus external factor, a terrible physician that doesn't use empathy, but only a paternalistic, powerful attitude to patient, and so on. And many other factors, negative experience, a surgical procedure who went wrong, and so on. So therefore, the goal is how can we understand the mechanism of placebo effects? At the beginning, one of the most powerful way to study, placebo effects was pharmacological conditioning, something that Gustavo did also a lot. So at the same time, while we were doing conditioning with apomorphine, in Germany, Gustavo was doing a conditioning with cyclosporin A, a drug that can inhibit the immune system. We use a drug that can facilitate the release of dopamine in the brain, a dopamine agonist. So apomorphine, dopamine agonist, was given 3 days before a surgical procedure. And then we move our patient with Parkinson that didn't respond to any pharmacological treatment to the surgical room. So the setting was meant to induce, a relief of their symptom during the surgical procedure. So the surgeon proceeded with insertion of an electrode in this area of the brain, the subthalamic nucleus. And so we use one of the 2 nuclei as control and one as target. And we had a big ambition question, like, okay, people who feel better, tell us, I feel better, they have less tremor, bradykinesia, rigidity, but also, is there something in the brain changing? We are in the basal ganglia, the part of the brain modulating motor movements. And so we were able to recognize this nuclei that is quite small, the size of a bean, because the firing here is characterized by this frequency, but also by bursting activity, as compared to the thalamus that is more silent, and here the substantia nigra that become with higher frequency. So we record many neurons, and when we ran this experiment, we asked our patient, Parkinsonian patient in the surgical room, how do you feel? And we found 2 distinct responses. Some people felt better, and they told us. Also, a neurologist came into the surgical room and assessed rigidity to the wrist. And so the scale that they use is the UPDRS. And this is the UPDRS reduction, the self-report of benefit, and this is the offline analysis of many neurons. This is just an example in one patient. So the average of all the frequency of discharge decreased after a placebo given after 3 administration of apomorphine. However, some patients didn't respond. They didn't perceive any benefit. The neurologist didn't find any change, and we didn't detect a significant change in frequency. In 2004, this was very exciting, or at least for me, as a junior person like you sitting in the room 20 years ago, to hear the spike of a neuron changing after a placebo administration made me think this is my area of research. And it was what we call an epiphany. This is what I want to do, because to me it was something like, wow, why a placebo that was saline solution injected to the forearm can change the frequency of the neurons. Of course, also the other part was interesting, the clinical, the self-report outcome, but to me, the brain mechanism was the most appealing. So, at the same time, we knew that placebo analgesia is related to the release of endogenous substance in the brain. So, in this case, again, we use a pharmacological conditioning. We use morphine. And we know that morphine is one of the most powerful pain therapeutic in our hands, right? So we invite health participants, like you again, to come to the lab, and we use a sphygmomanometer to make the arm painful. So the device, we measure blood pressure, you inflate to the maximum level to the forearm, and if you want to try, after 10 minutes, your arm will have paresthesia. That is very similar to what our patient with neuropathic pain feel. But also the arm becomes purple, temporarily, is reversible. So we invite health participants, they tolerate this kind of tonic pain only for 10-12 minutes. But if we pre-inject morphine, the tolerance is above 20 minutes. So our goal was, okay, can now replace morphine with a placebo. So after 2 administrations of morphine before the pain challenging test, we replace morphine with placebo. Still, you can see we can increase tolerance. But of course, no as morphine, yet much more than the 2 control at the baseline and post-placebo. You don't see here, but then we pre-inject people with naloxone, high dose, the dose that we could use on the street to somehow reverse intoxication from using drugs on the street. And we block these effects. It's here, beyond the translation. So what that said, oh, actually it's here. The yellow. So you can see that we block completely placebo analgesia. So what this tells us, when we observe this sort of powerful reduction of pain through a placebo preconditioning, at least for pain, this phenomenon is routed in the release of endogenous opioids. In fact, if we use an naloxone that block the receptor in the brain, we can reverse this phenomenon. So today we know very well that opioids are prevalent in the brain. We block placebo analgesia with high dose of naloxone, and this phenomenon is related to the release of endogenous opioids in the brain. Today we still try to use this kind of design to taper opioids, to somehow face the epidemic of opioids in the States. So I know that here it's less an issue, but in the United States it's the #1 cause of death. People who get prescribed opioids to manage their pain after a car accident or even a removal of wisdom tooth, then they become addicted and they challenge and sometimes they die. Actually, many times. So the goal was, can we use this mechanism to taper opioids, to somehow release endogenous opioids to help to get out of opioids? And we compare, conditioning 1 day apart versus 1 week apart. And you can see a placebo after morphine is almost as effective as morphine. So I hope that scientists doing animal research or human research or working with patients can expand that line of research. Another mechanism that is intriguing is the agonist mechanism. So I mentioned to you the apomorphine begin somehow an agonist, but also vasopressin is an agonist. So vasopressin is well known in neuroscience because together with oxytocin regulate our affiliative behaviors, our getting in love, our being friendly with a classmate. So we release these 2 hormones in a dimorphic way. The females in the room, or women, release some vasopressin differently than the men in the room. And there is a reason, and many animals across species release both oxytocin and vasopressin to regulate social behaviors. So our question here was, can we actually increase in the brain vasopressin and oxytocin to increase placebo analgesia? Again, we invite health participants to the lab. We randomize them to no treatment, oxytocin, low dose, vasopressin and saline solution, because even a spray of water in the brain does something with reduction of cortisol. And so we found that dimorphic effects in women, vasopressin increased placebo analgesia compared to all the other treatment and no treatment. These effects of treatment related to vasopressin was not observed in men. And the reason why this happens is because we can modulate the release of cortisol and change the level of anxiety temporarily, what we call dispositional anxiety. And you can see an inverse relationship. For example, if we look at cortisol here, the higher the cortisol, the lower placebo analgesia. Therefore, the goal is to try to understand more about the nature of expectations. And again, we came back to the lab to understand the mechanism. We know that we can expect something and get something else in life, right? And this is true also with clinical outcome. Patients come to us, to see us as physicians, and they come with a mindset. They want to feel better, no matter which symptom we treat. Anxiety, depression, fatigue, muscle pain, and so on. It is worthwhile to ask them, what do you expect from this treatment, from our interaction? And I show to you why. So we invite health participants to the lab. We had 3 different visual cues, red, yellow, and green. And we pair the cue that we call anticipatory cue with 10 seconds of painful stimulation to the forearm. This is like, if you know the Bialetti coffee machine from Italy, it feels like adding to the forearm something hot. And they had a remoter in their hands to block the delivery of stimulation when they were not able to tolerate pain anymore. And also, we had different phase. When the painful stimulation, this is visual analog scale to measure pain, was high, so the level was 8 out of 100, the face was fearful. When it was moderate pain, the face was neutral, and when it was low pain, the face was happy. So after learning that through direct experience, we moved to the scan, and we told them, today we do the same as yesterday. But what do you notice here? We change all the pain intensity to the same level, medium level of pain. But also we mismatch the anticipatory colors with the face. And the goal was, okay, when we mismatch what we learned the day before, if we learned that red comes with high pain, what would happen if we change the level of intensity to be moderate for everyone? And so if they were continue to see congruencies between the visual cues that they had learned the day before, they responded to the red plus fearful face with high pain, despite the level of pain was the same as the yellow intensity. And the same, they perceive, the green-related stimulations as low painful. We would call this as placebo analgesia and nocebo hyperalgesia. However, when we mismatch what they had learned the day before, we block placebo analgesia and we reduce nocebo effects. At the level of the brain, this was paralleled by an activation of the supramarginal gyrus and the angular gyrus. What that suggests, we can manipulate our expectation to feel more or less pain, and also this is corroborated by brain imaging. At this stage, we had studied placebo in the lab, mostly with direct experience, conditioning, or verbal suggestion. In 2009, we decided to introduce the transfer of analgesia through observation. If we watch someone else receiving a treatment that is analgesic, can we transfer also the analgesia to the observer? This was the setting, very basic again, very simple. There was a demonstrator receiving painful stimulation with red, no painful stimulation with green. And we told the participant that these electrodes, every time was active on the middle finger, would have blocked the transmission of the electrical shocks. After 12 minutes of observation, we move to the testing of our observer. And what you can see here, we assess all the pain at the high level of pain. And the goal was, if now they receive high level of pain with both green and red, would they still feel analgesia? And surprisingly, these were the effects. Merely observing analgesia in someone else transferred this level of reduction of pain to the observers. In terms of brain mechanism, we were curious to see what is relevant, why we learn, pain and no pain through observation. Of course, there is also the transfer of anxiety and pain. In fact, at this stage, we asked participants to lay down, watch the video where a participant was receiving painful, no painful stimulation with a cream that was sold as control and another cream described as a powerful painkiller. In reality, both the cream were placebo. So when they observe someone else receiving a therapeutic treatment, in this case, the green, they start activating both the temporoparietal junction, left and right sides, and that was their interpretation of what they were observing. But what is intriguing, they start modulating the amygdala as if they were receiving pain, and we had not even put the thermal stimulation on their forearm. And also they stimulate this area that we describe as sort of hub to accelerate or inhibit pain, the periaqueductal gray. So just to see someone else receiving pain, no pain, and the treatment engage in all these, brain responses. When we test them for pain with identical level of intensity, we observe an activation of the dorsolateral prefrontal cortex. And we use this as a seed to compare the connectivity between dorsolateral prefrontal cortex, temporoparietal junction. And this connectivity between what they expect and what they observe and learn through observation mediate placebo analgesic effects induced by observation. Interestingly, this occurs very fast in the brain. We manipulate and change the paradigm to study EEG response, electroencephalography, and we found that this response occurs within milliseconds, from 200 to 400 milliseconds. So we are good learners. We learn very fast to interpret the world around us, including analgesia. The next question, and this will be the last part of the talk. Many times I told you this experiment was conducted in health controls, people like you, like this audience, coming to the lab, volunteering, receiving their compensation and being with us for a couple of hours. So our goal was, do we see the same amount of magnitude of placebo effects in patients with chronic pain? We choose orofacial pain because it's a prevalent disease, orphan, there is no agreement how to treat this, and prevalent again in young people, so affect the youngest. So, and when we started this research in 2005, the prevalence was 5-12% in the States. Currently, jumped from 12 to 30%. So we truly need to do something for orofacial pain. So same paradigm. They came to the lab. We recruit from the street. We like to engage with the community. We have our flyers in the bus. We go to community event. So the first thing we offer is a 4-hour diagnosis of their pain. And then they walk for 2 blocks to our lab. And we do the same identical paradigm with painful stimulations. We also assess expectation. And the goal is to try to understand. They do respond to conditioning, as I showed to you, as well as controls, match for age, race, and sex. And this is the distribution of our participants. We study 802 participants, 400 controls, 402 TMD. And you can see it's very similar. So the magnitude distribution of placebo effects in chronic pain patient is similar to 12 controls. This is not surprising. Know that many trials fail when we try to develop new pain therapeutics because of large placebo responses. We have also people who are treated with a placebo and they respond with hyperalgesia. Unfortunately, it's the minority of our participants. Also, we try to understand which factors somehow can influence participants. We are lucky to be in a very nice ecological setting to study race, ethnicity difference. Baltimore welcome diversity. as of today, we have been studying Afro-American black versus white, but we are expanding to Asians and Hispanic people. We recruit in the lab 2 people from South America to try to study placebo effects now in the Hispanic population. For now, we have only the comparison between Afro-American black and white. So when we have a concordancy between the race of the experimenter and the race of our participant, in chronic pain patients we have larger placebo effects. What that means? If an Afro-American black participant with chronic pain come to the lab and one of our lab member is Afro-American black studying this participant, the magnitude of placebo effects will be larger. It's also interesting that when we look at sex difference, right, women suffer with a proportion of 3 to 1 more than men of pain. And this is related to many factors. So we wonder also for placebo. So men have, no difference in terms of placebo analgesia if they are studied by a woman or a man. But when it comes to women, if the physician is a man, they perceive us more powerful and their placebo effects is larger. In terms of psychological factors, we also study these people in terms of personality traits and other mindset-related factors. So first, to be able to do that, we had to split participants into responders and non-responders. These are the responders in blue, and here in red, the non-responders, people who have a reduction of less of 20 points on a scale from 0 to 100. Although these are people that may still wish to have this benefit in clinical setting. Anyway, we had a very large battery of surveys. We offer, comfortable setting to sit there with us for over 1 hour to answer all these questions. They can take break and so on. And then we use a machine learning paradigm to, instead of focus on single surveys, to somehow identify the valence related to these surveys. And we found that emotional distress, reward seeking, and I will show here, fear of the pain and openness were critically, involved in summarize this over 1,000 questions. So we reduced the 1,000 questions to 4 domains. Domain that link to specific system in the brain, like the reward system, the catastrophizing, and so on. And what we found was, our goal was to study placebo effects, conditioning, and expectation. Let's start for expectations. Baseline expectations are much lower, people without hope, in those people who suffer with catastrophizing and fear of pain. And no matter what we do, they try the analgesia, they continue to have lower expectation, and after the test as well. On the contrary, people who are optimists, open to new experience and so on, they tend to have better expectations at the baseline and after the treatment. The main difference between health control and chronic patient was the amount of reward seeking. Patients suffering from chronic pain seek pain relief much more than an health participant who doesn't know what pain is about. In terms of placebo effects, fear of pain and catastrophizing were associated with lower magnitude of placebo effects and placebo effects with lower percentage of responsiveness. Emotional distresses, people who tend to be distressed all the time and anxious, have also lower placebo effects that extinguish faster and lower magnitude of responsiveness. So we identify at least some domain like emotional distress, catastrophizing that can impact placebo responsiveness. Also, we measure sleep over 1 month before coming to the lab and the night before coming to the lab in both health controls and patients with pain. And we found that insomnia, people who have clinical insomnia, not who don't sleep the night before because of the test, rather clinical insomnia, is associated with lower magnitude of placebo effects. And the same poor quality of sleep, people who wake up many times. And this is very common in chronic pain patients. So what we are trying to do now in the lab, identify the predictors of placebo responsiveness. Like if we are in this room, 50-60% of you respond, but some other people may not respond. And our goal is, how can we understand in advance who are the responders? We look at the 3 SNPs, single nucleotide polymorphisms, that have been published in the literature. The SNP for endogenous opioids, OPRM1, the SNP for endogenous dopamine, COMT, and the SNP for endogenous cannabinoids, FA. And we found that we compare with verbal suggestion and learning. There is not really an advantage to study 1 or 3 polymorphisms. Rather, we need to combine the genetic cassette with the manipulation we use to study placebo. So when we account for verbal suggestion versus conditioning, we have the best predictor of placebo analgesia, suggesting that we can rely merely on a SNP to predict placebo effects. Rather, this is a multifactorial phenomenon. We need to learn more about the history of this patient and so on. And continue on this omic biosignature and so on, we start and receive a grant to look at omic or transcriptomic profiles. The RNA gene expression is more sensitive. We need less sample size to study this phenomenon. So randomly we went to the freezer and select 54 of the 402 TMD, and then again 31 TMD for a validation cohort. We call this discovery cohort, and then we did the PCA to validate the genes identified in the discovery cohort. This volcano plot is very complex, but show that during the placebo responsiveness that we treat as a continuous, we have some genes that are downregulated, some genes that are upregulated. Then we focus on the top 20 genes, and we replicate the 6 new genes in the literature for prediction of placebo effects. We hope that other labs will follow up, and our lab will continue to do that, seeking new markers for understand more about placebo analgesia, the interaction with the immune system, and so on. And with this, I would like to conclude. I hope I convince you that placebo and nocebo effects are biological phenomena. Placebo and nocebo effects can be studied in both animal and human models. Animal, I hope Gustavo will tell you much more than I can share with you. And finally, I think the future would move forward if we are able to identify the responders and the non-responders, or at least those people who are likely to respond very strongly, and maybe they don't need this cocktail of pharmacological treatment that other vulnerable patients may need. That can also help with drug development as well as symptom management in surgical or clinical practice and so on. And I would love to thank my team. That was very fast. And of course, all of you for being here together today. Thank you.
Thank you, Dr. Colloca. You leave us a strong collection of lab data, and we must now translate them in clinical application in our country. Thank you, thank you. Well, now, ahora yo tengo la oportunidad, el honor de presentarles al doctor, ya no me voy a equivocar de tu nombre, el doctor Gustavo Pacheco-López. Él es un excelente amigo, un excelente colega que nos hace ya 15 años o un poco más, él nos empezó a compartir sus estudios sobre condicionamiento y placebo, como lo invitamos, lo trajimos a la Facultad de Psicología, y ahora es un sobresaliente funcionario de la UAM Lerma, y tenemos el honor de escucharlo y de realmente el privilegio de combinar lo que nos compartió la doctora Colloca con su experiencia. Te escuchamos, doctor. Bienvenido.
Muchas gracias por este amable recibimiento en la UNAM. Si pudieran regresar a la primera diapositiva, va a ser muy breve mi comentario. And I will switch to English because I would like to thank Luana for this wonderful presentation. And I hope that also for all the Mexicans, we should speak English. I mean, science speak English, and we must speak English if we want to communicate with scientists all over the world. So please, if you could return to the first slide, because I was really pleased by... hay una... La primera, por favor. Just, I want to... si pudieran evitar este distractor, porque en realidad nos deja inertes a los presentadores. Tal vez puedo hablar en español y en inglés, pero si pueden quitar la traducción, por favor. Carmides, Plato actually starts with the origins of medicine. And just read this nice side from Plato dialogues. I reply, Plato there is a dialogue between Carmides and then say, when he asked me if I knew the cure for the headache, again pain, 2,000 years ago, more than 2,000 years ago. I replied that it was a kind of a leaf which required to be accompanied by a charm, shaman. And if a person would repeat the charm at the same time that he used the cure, he would be made whole. But without the charm, the leaf would be of no avail. 2,000 years ago, we already recognized the powerful of our expectations. Los diálogos de Platón. Next, please. La siguiente. Okay. I will just make some, let's say, highlights of particular research that we perform in particular area of immune-related placebo responses. And almost 20 years ago, time flies, we published this seminal paper in which we tried to document some of our thoughts and the evidence that in 2006, we already had about placebo immunomodulatory responses. And Luana today confirms that still are valid. Many of those basic mechanisms, expectation and conditioning is still on the focus of placebo research. What we have done during those 20 years that are from that time, okay, con esto puede controlar, okay, this nice paper that we published almost 2-3 years ago summarized the state of the art of placebo conditioning on immune functions. There are several parts of the immunity that are modulated by expectations, but particular by conditioning. Immune system is well equipped to communicate with the with the central nervous system as well as the neuroendocrine system. So I will not go through all these details, just to summarize. There are evidence on transplantation medicine, allergy, autoimmune diseases, metabolism, and cancer, that this interaction between the brain and the central nervous system is critical for the outcome. This is one of the maybe more significant papers that the colleagues from Germany have published, in which they try to demonstrate, or basically they demonstrate the translational power of learned immunosuppressive in renal transplanted patients. So using conditioning, it is possible to benefit the patients even in this critical stage of renal transplanted conditions. So maybe if you would like to check the details, you should go to the papers. We have also a review and have some evidence of the effect of conditioning hormonal responses, insulin, cortisol, growth hormone, and melatonin. Just to give you some examples, as well as oxytocin, we just heard from Luana some of the evidences. Okay, I will stop here trying to connect with the physicians and also with the practice of psychologists, utilizing placebo mechanisms for dose reduction pharmacotherapy seems to be one of the most promising avenues for utilizing the power of our expectations and also of our capacity of learning. And if you would like to hear me in Spanish, there was also a nice conference years ago organized by the Colegio Nacional, in which you could follow most of this research in detail. Thank you very much, Luana, for your nice presentations. Thank you very much for for your attention, and well, we can also hear from your questions and may try to comment on. Thank you, Benjamín.
Bien, vamos a destinar unos minutos para preguntas y comentarios que nos deseen hacer. Les comento que en la transmisión a YouTube sí se estuvo manteniendo los letreros de subtítulos en español para que nos podamos compartir con todos esta presentación. Quiero también mencionar que nos acompaña en esta conferencia el doctor Yavin Shaham. Él es del National Institute of Health y le damos la bienvenida a la Facultad de Psicología, doctor. Muy bien, pues si tienen preguntas y comentarios, aprovechen esta oportunidad. Vamos a destinar unos 10 minutos a esto para poder atender otras actividades. Adelante, les van a pasar el micrófono.
Hi.
Question for you and for you, Dr. Pacheco.
I wanna ask if the autonomic alteration can influence the placebo or nocebo effects, like in neuropathic diabetes or multiple sclerosis.
Thank you very much for the question. So multiple sclerosis probably is one of the diseases where we didn't yet invest in exploring how expectation conditioning can affect, the symptoms of multiple sclerosis. I remember when I was in Turin, I engaged several times with my colleague neurologists suggesting to do something, but we didn't publish, unfortunately, anything yet. But going back to the beginning of your questions, how autonomic measurement and autonomic responses, like the vagal responses or sympathetic responses or heart rate frequencies, can affect placebo, we did publish in that. In particular, we were manipulating during the intervention with deep brain stimulation in the surgical room, and the higher is the vagal stimulation, so that we can also achieve with, implantation of the vagal nerve, the better the placebo effects. So definitely there is a relationship between autonomic measurement and in particular vagal system and better placebo effects. But we do need yet to study sclerosis. Yeah. Thanks
Thank you
for
so much.
asking.
Hello. I ask if the placebo anesthesia can be activated only by spectatis or can be activated by hypnosis.
I'm glad that you asked this question because truly we were driving from a session at the conference, the AMED, where a person who does hypnosis spoke after me. And so hypnosis is seen as a different procedure than placebo, in the sense that if we compare the brain mechanism of hypnosis and, for example, conditioning analgesia, we see different brain mechanisms. Although this is a sort of simplistic answer that I'm providing to you, because of course the brain responds differently if I'm touched by worm or by cold, or if the brain detects hypnosis versus conditioning. So in terms of mechanism, clearly it's different. In terms of outcome, we can achieve similar and comparable outcome with placebo analgesia and hypnosis. And I let psychologists in the room, to comment on the psychological aspects of hypnosis versus placebo analgesia. What I learned today from my colleague is that hypnosis is based on active expectation. You change the mindset. You somehow do a work towards processing the pain. On placebo, we rely on expectation of benefit that can be seen as a more passive expectation, where you take a drug and you get better, instead of going through a process of education and remodeling thoughts related to pain.
Thank you.
Sure. Oh, thank you. Other questions?
Hello. Do you hear me or?
Sí.
Okay. First, my English is not the best.
That is perfectly
No,
fine.
it's not my best ability. So I am going to learn the translator. Okay.
Yeah.
First, I would like to express my admiration for your work. I really, if I am a member of Nobel Committee, I will have you on my list. Okay. Yeah, it's really impressive, amazing your investigation and the implications that it has. And my questions are going to the implications about your work in other field that is cognitive science. Okay. The question what I want is one question that I would like to ask you is if based on your experience of placebo effect at the level of neurobiology and behavior, at a cognitive level, it is possible to speak that the placebo effect is a problem of representation. Okay, this is the first question. Is at cognitive level, the placebo is a cognitive problem.
I understood. Yeah.
A representation problem.
Yes.
And the second question is, will you consider that the problem of representation of knowledge of cognitive science is still relevant not only at a cognitive level but also at neurobiological level? That's are my two questions. Sorry, I am so nervous.
Okay. Wonderful questions. So let me comment on your not just questions. First, thank you for commenting positively on my talk and the research we conduct. I hope some of the Nobel committee will hear your voice one day. Second, the English. Your English is good. I learned English as an adult, and I experienced the challenging. So I encourage all of you to, somehow push. It's a bias. We think that we can't learn English, but we all can learn. If I learn, you can learn. I truly didn't have attitude for languages, so continue, keep doing, learn English, so that you can communicate with the world and not just with the United States. And the questions that you raise are very interesting. Is placebo a matter of representation, a cognitive representation? There isn't so much research. Actually, Hispanic guy, Lugar, published about cognition and open label placebo, that are placebo given without any deception. So you offer placebo to participants, and he was studying the mindset. And there is another paper by Torweger that is in press coming out, I think in this few months coming, where again they study open label placebo in patients this time, patients with chronic pain. And actually it's a matter of cognitive representation. It plays a role. And the more we learn, the more we understand how cognition becomes relevant. Yet, phenomena like the one that I mentioned with conditioning, with, for example, apomorphine or with cyclosporin A that Professor Gustavo studied earlier on, this seems to be more independent than cognitive processing. Like, I don't have to believe if I'm exposed to apomorphine, or cyclosporin, the neurons change in the brain. The interferon gamma and IL-6 change in the blood. And if I tell someone, Gustavo, now your interferon gamma will go down, this doesn't do anything. My interpretation, and I published this in 2011, and now other people are starting hearing this, is that for symptoms and behaviors that we have awareness of, like I know what is reduction of pain, I know what is the headache that we learn from Plato, I know what is anxiety or fatigue, but I have zero idea when my interferon gamma change. I don't know what my IL-6 is doing in my body. So to change that, the cognitive representation doesn't help. I can do hypnosis, mindfulness, and I don't see a direct impact on, humoral changes, blood markers. On the contrary, we see immediately an effect on anxiety, pain, fatigue through cognitive representation. And the only study that is closer to that we done in our lab and we published recently is related to yogis. So among our 802 participants, some were professional yogis, some were practicing yoga daily, and these were people that somehow develop interoception to an extent that those who don't do yoga don't have. So these people tend to have lower placebo analgesia through our manipulation with high-low pain. And they were telling us, you change the intensity of the pain. So they had a representation of their body that is different than someone else who doesn't do yoga daily. So they perceive that we change the level of pain, and so our paradigm didn't work. And that was true also in people with psychosis, schizophrenic patients. They realize the difference. So when we switch to all the painful intensity identically to study conditioning, they actually get this body representation. They get this body change in terms of perception. So I hope I answered your question. There are many more experiments and research to be done in this field. It can be very interesting to learn more about how cognition, body representation can impact the ability to produce placebo effects.
Thank you.
I will take the opportunity, Luana. What about different age groups, so elderly people and children?
Yeah.
Yeah. What about age groups?
Yeah.
Elderly and children, because most of the data comes from young, healthy
Adults.
adults, and also in the case of patients,
Yes.
really few children studies.
So does age influence placebo effects? We know that brain development is very good and very important, but also brain aging is a reality. even sometimes you have some Biden effects, you know. And we refer to that to some moment where you are not brilliant as usual, you know, this happened to everyone. So this can be temporary, you know, you didn't sleep well the night before, you are jet lagged and your brain is less effective. But also aging is a reality, we all get old. So our lab starts inquiring, you know, is there any difference related to age? And it's interesting, we didn't publish yet the results, we are writing a paper. There is a U curve. The best placebo effect is observed in the youngest and the elderly. Middle age, like me, we are the worst. I don't know the explanation, but when we age, I mean, I was telling this to some colleague in Paris at INSERM, and they explained me that when we age, we use our wisdom and cognitive resources that we don't do while we are busy with our daily activity in middle-age life. And the same way, kids, teenagers are the best responders. And there are some papers show that the higher the magical thoughts, talking about suggestibility and so on, the higher the magnitude of placebo effects. And we know that we kiss a child and he or she feels better, right? Or an hug can be so powerful and analgesic. Somehow we lose that when we grow. Our middle age is the most vulnerable age to somehow begin less a placebo responder, but also more vulnerable to chronic disease. Fortunately, while we age, we go back with the U curve to higher placebo effects. Yes.
Si alguien quiere preguntar algo en español también.
There is another question here.
Last question.
Two questions.
Yes. Thank you. I'm glad to meet you in person. I'm very impressed with your work, and I would like to ask if with all your data that you collect in this time in your research, do you think that with these tools in AI, machine learning, some analysis like that, we can use the data to predict how the effects of the treatment of placebo could affect this that you say, no, that the middle age, we don't know why, no? But maybe with the new techniques in this kind of analysis, we can predict or we can make hypotheses of why. Because you have so much, a grand amount of data that we can use these tools to predict some in the future.
Yeah. Thank you for your comment. So AI is powerful, it's growing around us. We can have an attitude that is positive and accept and endorse or be skeptical. Either way, it's a reality. AI is changing the way we see data, the way we interpret the world around us, and also the way we do clinical practice soon, you know. I serve as a reviewer in many different countries for grant writing, and I see that it's coming. And so I don't expect that we have to wait too long to see the first results with AI and placebo responsiveness, placebo predictors. All our data are based on NIH funds. That means public available. You contact us and we can share our data. There is an interest in our lab to try to use machine learning. There are a few PhD students that are investing in that direction. How can we interpret this data set? But also we are working toward the consortia with other labs like Germany. Gustavo knows is very prolific with placebo research. And I hope this talk will inspire more collaborations. Also in Mexico City, you have 20 million people here. I mean, we are a small village compared to you in Baltimore. So any person who would like to collaborate, there are ways to do and work together. So I truly hope that this is just a seed to stimulate more science and more research. Clinical based, basic based, doesn't matter. Yes.
Thank you.
Yes. And another question here. Oh, back?
Otra pregunta.
Yes, there. 3 more questions.
Thank you very much for this wonderful presentation. It was really very enlightening and brings a lot of hope. But I wanted to ask you something that it's not as related with your knowledge, but as related with your experience. Coming from a country where pharmacotherapy is so on the top of everything, have you had any problems trying to talk about why we don't need that much?
Yeah. So I'm a physician scientist, not a psychologist. So the first problem was myself, because I was very skeptical about this phenomenon when I start my PhD. I told my former mentor, I'm a physician, not a psychologist. I want to do things with really drugs and stuff like that. So I was the first one biased towards that. And we know why, because when you are trained as a physician, psychology is just a course. There, I got 1 course in psychology in my 6 years of medicine. But also when I started writing a grant, as of today, 20 years of research, I received 0 grant from drug companies. No one is interested in the research I do. Fortunately, the National Institutes of Health, that is our federal agencies for funding lab, somehow like my research. I'm very, grateful forever to them. Begin train as a, senior fellow first for 5 years, and then begin funded as a PI. So without them, all this will not have been possible. There is this obstacle. I perceive this morning. Today here, I feel much more rewarded to talk to young people than to talk to physicians in the other audience. They were doing their business. They were not even listening to me the same way you are listening to you here. Why? Because there is a structural bias. We are educated to separate things, psychology, medicine, pharmacology, and unfortunately until we break down these walls, and we know our walls are terrible, and I'm not a politician here, but we don't believe in walls of any nature. Walls can be mental, can be separation between countries in the same country. No walls. Once we break these walls in our mindset, we can progress and incorporate psychology with pharmacology and medicine. And the challenge is when we use psychopharmacology to condition, to reduce the reaction to a kidney from a donor through psychopharmacology. This shows to us that there is so much to do yet. And psychopharmacology, I think, will be the future, because the more we learn about the immune system and the connection between drug and the immune system and psychology and cognition, that should be truly the way to go, I think, today. Yeah. And another question on the back there.
One more question.
I see a person there. Yes, you. You can use your voice. We can hear for sure. I can hear.
Okay, thank you. I know that. Okay, thank you so much. One of my questions is, what were the limitations that were presented through all that studies, and that what are we expected to be changed in the future? And I know that it's very different in each context. But now, talking about here in Mexico, that we are expecting that we can be more spread in our, maybe in our school, of course, and that I would like to know more about the limitation and that what are we expecting in the future.
Yeah. Let me see if I interpret well your answer. What we can do as Mexican people for the future in clinical practice, but also in education. We mentioned today the needs of, advancing this science, but also we want to translate this science. Translation is not merely learning that empathy, placebo are neurological based, biological based. Rather, will be to reach out to this, education, how we train the next generation of physicians. And eventually this next generation of physicians will be able to implement what we are learning. There are yet some barriers, one is calling this placebo research, because already we cut a lot of people who may not have an interest in non-pharmacological approach, although placebo may be even a drug, right? So somehow to think outside the box, how can we elevate this science to reach out to as many people as possible? And I think speaking to people like you is very relevant, because I have an idea or a set of ideas, and you may be brilliant, having a better idea than my ideas, and reach out and say, hey, Luana, can we sit together and talk? So don't be shy, reach out to people. You have an idea, you want to do something. Come to my lab, come to the States, move away, and bring back what you learn, like Gustavo did to your country. It is very essential to, be curious, challenge yourself to do more, because all this come from small initiative. I can share about my life. I was born in a small village, 3,000, inhabitants, and I didn't envision all I have been done. But every time there was an opportunity, I was not afraid. I didn't turn any opportunity down. I said, let me try. So you should do the same. You have an idea, you have an opportunity. Seek resource, talk to the chair, talk to your faculty, and ask, I want to go and spend 6 months in a local lab and learn more. They will contact me and they will arrange. Physicians, you are the most empathic people I've ever met. It's my first time I came to Mexico, but the way you practice clinical, interaction doesn't exist in other countries. In Baltimore, my colleague physicians are in front of a computer typing while they talk, literally like that. They don't even engage with eye contact with their patient. So we needed to reinvent the wheel and tell them that empathy is relevant. But they don't know, they don't practice these things. So then we needed to raise this cultural difference and learn from one another. You smile, you laugh, you interact with meaningful faces. That is powerful. It's already a strong placebo trigger. So now the point is, how can you elevate what you do naturally with rigor of science to teach other people, other country, or maybe expand within the Mexico country what you can do? Can you somehow structure this kind of approach to medicine to become powerful, to the bedsides, but also to other people as an example of education? Yeah.
Thank you so much. And I hope that maybe in the future, maybe this school and maybe they can provide us many tools, for example, to, get more curiosity and all that. Thank you so much.
I want to offer. The book that was the last slide, it's available for free, so you can download it for free. We invest intentionally. We pay the license to have everyone the ability to download and read the book without spending $70, I don't know what is the price, so you can read it for free. But also at the center we have at University of Maryland, the Placebo Beyond Opinion Center, we offer lectures, again for free. Once you become a member, also for free, you can attend our series, and maybe you learn something that become relevant for you. Be curious and practice English. That is our language for science. Yeah.
Italian too.
No, come on. Thank
Bien.
you.
Le agradecemos mucho a la doctora Colloca. Les comento que hemos estado en un congreso médico desde las 8 de la mañana y la doctora ha tenido 3 conferencias y en todas ha estado muy...
Good use of my time, or otherwise.
Bueno, yo quiero agradecer el apoyo de nuestra directora doctora Medina-Mora, verdaderamente invaluable y tremendo, un respaldo que nos ha dado, y a todos los colegas que nos apoyaron para esta sesión que para nosotros representa el enriquecimiento de la caja de herramientas de los psicólogos del futuro. Los psicólogos del futuro van a tener en su cajita de herramientas el placebo y todos los estudios de la doctora Colloca para intentar paliar, para intentar apoyar a la cantidad de mexicanos que tienen problemas serios, incapacitantes, costosos, de dolor crónico asociado al cáncer, de dolor crónico asociado a problemas diabéticos, de neuropatías. Por cierto que la traducción no traduce bien neuropatía, pero bueno, ya le iremos enseñando. Y de los problemas relacionados con las condiciones cardiovasculares que nos afectan a muchos de nosotros. El placebo y otras herramientas no farmacológicas que son principalmente deberían ser manejadas por especialistas en psicología, es una herencia, es una donación que nos hace la doctora. Su libro, por cierto, es gratis, ya no se pudo ver, pero gratis.